At the cytoplasm, it's going to encounter a ribosome. And on this diagram, this kind of big rectangle thing here is the ribosome. Now the ribosome has two subunits, or two parts to it. It's called the P site and the A site. P stands for polypeptide, or a chain of amino acids. A stands for amino acids. What is going to happen is the ribosome is going to hold our messenger RNA, which is right here, our single-stranded mRNA, and help the cell to decode the message that came from the nucleus, from the DNA. The process of decoding this message is called translation. So translation is decoding the DNA message and when that message is decoded, it's going to make a protein. And proteins are what build your body structures, as we already discussed during biochemistry. So our last step of what DNA can do is called translation. All right, we'll take a close look, or quick look here at how translation works, and then kind of get it out to bigger picture idea in a minute. So first of all, the place on the mRNA where the ribosome always binds first is a place where it says AUG. It's our start codon. AUG. Oh, we're back. I'll put it here. AUG. Because remember, this is the P site and this is the A site. So it's going to bind to the AUG of the messenger RNA at the A site. In order to get an amino acid into place, though, we need the help of something called a tRNA, or a transfer RNA. Okay? The tRNA holds the opposite of the AUG. So the opposite of AUG is UAC. So the three-letter patterns on the mRNA are called codons. So this is a codon, C-O-D-O-N. And the three-letter segments on the tRNA are called anticodons. Now the anticodon is just there to make sure that it can get to the right codon, because they match up together, A with U, G with C, so forth and so on. But what it wants to match up is what's at the top here, and our oval that's at the top is an amino acid. In this case, the amino acid that AUG codes for is MET, or methionine. And hopefully you remember that there's a chart that you use to figure out the amino acids, which I'll show you again in a minute, so you don't have to remember all of them, but you should know off the top of your head that the first amino acid is always methionine. Alright, so what's going to happen then is the transfer RNA is going to carry that methionine into the right place, like that. So the AUG is going to match up with the UAC, bringing the methionine into the A site, and then the entire ribosome is going to move over. Okay. Now we have a new amino acid in the A site. The new amino acid is going to be new codon. The new codon is going to be CAG. The anticodon for CAG is GUC. We need to read the message down here, the CAG, to figure out what amino acid comes with GUC. So we're going to take a look at our chart real quick. Pull this down. We want to find GAC on our chart. It looks like GAC is right in here. Aspartic acid, or ASP, is our abbreviation. We'll push this back, and we'll write that in. Not with that marker. ASP. So aspartamine is the amino acid that's coded for by CAG. So the GUC anticodon on the tRNA will bring it over to the right spot. Let's move our next anticodon with the amino acid over. Okay, now that we have two amino acids, we're going to need to bond them together. So we're going to do that with a covalent bond, and then the ribosome shifts over again. So now this P site up here is holding the growing polypeptide chain. The A site always gets a new amino acid. Let's do another one. Let's say this next one is UUA, which means the anticodon for UUA be AAU. We need to check out what the amino acid is for UUA. Let's highlight it for you. This one up here. UUA stands for leucine. Put it back. Oops. I'm going to write in Lu for leucine. And it's going to go in to match up with the mRNA codon. There we go. Okay, the leucine binds to the aspartamine, the covalent bond, and the whole ribosome, moves over. There we go. Okay, this process keeps going until we get to something called a stop codon. So let's take a look at what that is. If I pull my tab out again, you can see that there are, get it away from all my writing here, there are 3 that are in red. Okay, right here, here, and here. Those 3 that are in red tell the mRNA to stop decoding at that point. So we're going to do a UAG in order to stop our coding. UAG. On the tRNA anticodon, UAG codes with AUC, and again, that's a stop codon. It does not code for an amino acid. Okay, we'll put that in the right spot. It's up to here. We still bind our 2 amino acids together, and then the process is basically done. So when it's all said and done, the ribosome is going to go away. Go away, ribosome. Put it over there. There we go. The amino acids here are going to end up being longer than just a chain of 4 amino acids. There's going to be hundreds of amino acids. That is going to be our protein. So maybe it's the protein that makes your eyes blue, or it's the protein that makes your hair straight. Whatever it might be, that's our protein that helps to build us. The tRNA, which is down at the bottom here, okay, this anticodon that's left over, gets put back together with other amino acids so that it can be used again. And the mRNA, down here, can get read again or decoded again to make another protein just like what we had up here. So you can make lots and lots of proteins from one mRNA strand. The ribosome, down here, okay, can get reused again to read that new mRNA strand that gets recoded. So all of this basically is recyclable, is what I'm trying to say. So the protein leaves the cell to go do whatever it's supposed to do, but all the other parts, the anticodon, the mRNA, the ribosome, all get reused to make more protein. All right, a little bit closer look at the tRNA, just because I think it's worthwhile to see what we're really talking about here. The tRNA is made of RNA. So RNA, remember, is single-stranded. And if you follow the letters in this diagram here, you'll see that they're all letters we've talked about. A's, G's, C's, and U's. And they're folded in a way to give tRNA this kind of, I guess, X or plus sign looking shape. At one end of this shape, so down here at the bottom, okay, there's a set of 3 letters. Do you see these down here? That's our anticodon. At the top of the plus sign is where the amino acid actually attaches. So when we say tRNA has 2 parts, okay, it has an anticodon and it has an amino acid. That's kind of simplifying things a little bit because it is a pretty complex molecule in and of itself. All these little dots in here represent bonds that are holding the strands of RNA together in this shape. Again, they're hydrogen bonds. Okay, now we'll go back out to big picture and put all these steps together that we've been talking about. So the purple part on your diagram represents the nucleus. The kind of tannish part toward the bottom is the cytoplasm. In the nucleus, we've got a big egg-shaped molecule, which represents RNA polymerase, making a red strand of mRNA. Now remember, that process was called transcription. So we're going to write that in there. So this first part is transcription, making the mRNA. The next thing that happens is the ends of the mRNA get either that 5' cap or that poly A tail. The introns get taken out, and then the mRNA can leave the nucleus, which is happening right here. We call that, again, RNA processing. It's all a review of what we've talked about already. Once the mRNA gets out of the nucleus, oops, went too far back. Okay, then scroll down a little bit here. It finds a ribosome. Okay, so the brown kind of structure here and here comes together around our mRNA to hold it in place. These teal-looking structures are the anticodons, so the tRNA anticodon. The little purple structures are the amino acids. So up here, what's happening is the anticodons are getting stuck to the original or the correct amino acids. Once that happens and our tRNA is formed, again this is tRNA, then we can start translation. Translation, remember, is reading the messenger RNA, this red ribbon of stuff, to make a protein. So this first one would be AUG, bonds with the anticodon UAC to bring in methionine. The methionine amino acid is always the first one, so that one would be here. Here's the second amino acid, third, fourth, fifth, sixth. So this is the P site. At the A site, we're bringing in a new anticodon with 3 bases to match up with the 3 bases on the mRNA, and it has another amino acid ready to attach to the protein chain.